Okay, ladies, so this video will cover a portion of your notes that I think is not quite as hard to understand. Um, we're going to look at the structural background on bacteria and viruses, and then also how they actually infect you. And I think this is, if you hear it, I think it'll make sense. And then we'll spend our time in class talking about how your immune system works, which is a bit more complicated, and then how specific infectious disease agents give you a problem, which is a lot more exciting. So if we start with bacteria, we talked a little bit about prokaryotic structure back in the first semester. And in fact, I think we saw this picture before. So we're going to recap that and then add quite a bit of detail to the structure of a prokaryotic cell. When you look at the inside of a bacterium, it's extraordinarily simple. There's no nucleus. Again, that's what makes it a prokaryote, right? But it does still have DNA, and it has DNA in an area called the nucleoid region, or the nucleoid. This is not really like a defined space. Basically, wherever the DNA is located is called the nucleoid, but because there's not a nucleus, there's no membrane around it or anything like that. Now, for most of these bacterial cells, the genetic material is just a single circular chromosome. So it's still double-stranded DNA, but basically they just have a single circular loop of double-stranded DNA inside their cell. And then it might be all tangled up like you see in this picture here. Now, if we look at the rest of the internal structure of a bacterial cell, you might remember that there's really not any other membrane-bound organelles in there. There's no mitochondria, there's no chloroplasts, there's no lysosomes, nothing like we see in our eukaryotic cells. The only structure they do have is ribosomes, right? Remember, because everything needs proteins. And so just like you and me, bacteria are going to need some kind of ribosome. The difference is that they have a little bit smaller ribosome than we have, and we call it a 70S ribosome, and so that might sound a little bit familiar from first semester as well. Now, one other unique thing about the inside of a bacterium is that a lot of times it has extra DNA. We call this extra chromosomal, right? Because it's separate from the primary chromosome or the primary set of DNA that a cell would have. So in a bacterial cell, we see a single circular chromosome, like you see here. Then we have this extra piece, or sometimes extra pieces, of DNA called plasmids. Now these are also circular, so they're loops, and these are also double-stranded DNA, but they're usually smaller, like the picture shows you, than the bacterial chromosome. And what's really cool about a plasmid is you can make copies of it, and then you can pass those to another bacterial cell. We call this process transformation, a process in which one bacterium can transform to become like another bacterium. Right? So in other words, it's kind of like we pass a little note of information from one bacterium to another, and now the new bacterium can take on those characteristics. Whatever genes are located on the plasmid can now be expressed by that new bacterium. So this is a little weird or silly or something, but let's say that this bacterium up here is able to make Pepsi. Okay, so it has enzymes that allow it to generate Pepsi inside its cell. If the genes that tell that cell how to make Pepsi appear on this plasmid, and we copy the plasmid over to a new bacterial cell, now this bacterial cell can also make Pepsi. And so this is what bacteria will do, is they'll pass information with these plasmids. A lot of times a plasmid will carry a very beneficial gene. And one of the most well-studied of these beneficial genes that can get passed between bacteria is a gene for antibiotic resistance. Now we'll come back to this more later, but antibiotic resistance is a serious problem for us as humans, because what this means is that our medicines, antibiotics, don't work. Right? If the bacteria can resist an antibiotic, it means our drugs won't kill it anymore. So that's scary. And if one bacterium knows how to resist our antibiotic, and it can share this information with the other ones, you're going to have a bunch of bacteria that are really dangerous. Now, this is a good opportunity to talk about or mention reproduction in bacteria as well. Remember, they do not reproduce sexually like a multicellular organism would, but they reproduce asexually, meaning that one cell simply splits into two cells. It's kind of like mitosis, but this is the whole organism that does this, right? So this is an asexual type of reproduction called binary fission. Binary means two, and fission means to split. All right, so split into two, binary fission. Because bacteria don't have sex, they don't share genetic material, right? They don't have that genetic variation that we receive through meiosis and through sexual reproduction. What they do have is a process called conjugation, in which they can transfer DNA from one cell to the next. So this is what we were just talking about on the last slide, is these bacteria can pass DNA from one cell to the other, and that allows them to transform. So the process, like the physical process of conjugation, leads to transformation. Right, so if two bacteria conjugate, then it'll transform them and they'll take on this new piece of DNA. Now, the external structure of a bacterium is a bit more complicated. When we talk about eukaryotes, you've got a cell membrane. Sometimes you've got a cell wall if we're talking about a plant. Bacteria are different. Bacteria have a much more complicated external structure. And when we look at that external structure, we can break bacteria into two groups: the gram-positive bacteria and the gram-negative bacteria. Now, here's what this means. The gram-positive bacteria and the gram-negative bacteria will react differently when they're exposed to a certain kind of stain or a certain dye and then looked at under a microscope. So we talked about, staining our tissues back before Christmas. Gram stain is one of those stains you could use. Now, if your bacteria are gram-positive, they are bacteria that will take on the full stain and have a darker color. So they're going to appear as a dark purplish blue cell under a microscope. A gram-negative bacteria will reject the stain, hence the term negative, and they'll just kind of slightly get tinted by the stain, so they have a much lighter, kind of a reddish pinkish color to them. So let's look at some pictures. So on this slide here on the left, or this picture here on the left, these round cells hopefully look a little bit familiar to you. Those are white blood cells, right? So this is like a picture taken from a human tissue, and all these round cells that I'm now crossing out, those guys are white blood cells. So what we leave are these really dark purple rectangles, right? So I'll kind of circle some of them here in blue. This whole group here. Now, every one of these dark purple rectangles, so when I'm looking at this here, I'm going to break this up. See, these are all separate cells. You can kind of see where they separate from one another. Each one of these is a bacterial cell, and this is not something that you'll have to know, and I'm testing you on, but these are gram-positive bacteria called Bacillus anthracis, and so you might have heard of anthrax before as a sort of actually a bioterrorism way. You can anthrax in powdered form you can put into an envelope and potentially make some people really sick, and so there were some scares back in the 1990s and early 2000s that people were sending anthrax in the mail to different people and stuff. But this is actually a bacterium called Bacillus anthracis. Causes a pretty nasty illness, but for our purposes right now, I just want you to recognize that gram-positive is going to give you this dark purple color. Now, the opposite thing is gram-negative. These gram-negative cells, which is everything that's in this picture, these gram-negative cells have a different external structure, as we'll see in a moment, so they sort of reject the stain. Rather than becoming dark purple, they just sort of get tinted a little bit of a reddish pink color. This is a bacterium called Pseudomonas aeruginosa, which is fun to say, but you don't have to know that either. Just recognize gram-negative, going to have this lighter red color to them. Now, the question becomes, why do they stain differently? And it has to do with the structure of the outer portion of the cell. So, when we look at the outer portion of a bacterial cell, we call it the cell envelope because it'll actually surround the cell and maybe have several parts to it. So whereas we just have a membrane on our cells, they're going to have a couple different things. So we call it a cell envelope. Every bacterial cell has an inner cell membrane made up of phospholipids, proteins, a little bit of carbohydrate, just like ours. Okay, so they have an inner cell membrane at the innermost part there. Outside of that membrane, all bacteria also have a cell wall, and the cell wall is made out of carbs and proteins. Now, in bacteria, the major component of the cell wall is a structure called peptidoglycan, which is actually sort of a mix of a protein and a carbohydrate structure all put together. Peptidoglycan is unique to bacteria and prokaryotes, so we only see this peptidoglycan material in the bacterial cell wall. Now here's a picture that breaks down the cell envelope and the differences between gram-positives and gram-negatives. So here on the left, we've got our gram-positive cell envelope. It starts with an inner cell membrane, here in yellow, and then it has this very thick cell wall outside of that. Now, this really thick cell wall can absorb a lot of gram stain. So you end up with this really dark purple, right? This is the one that turns out dark purple color because the cell wall soaks up a whole bunch of that stain. Now this is the opposite of the gram-negative example here on the right. Gram-negatives, again, start with an inner cell membrane, here in yellow. Then they have a very thin cell wall, followed by an outer cell membrane, another layer of phospholipids. Now, the outer cell membrane is what sort of resists the stain. And the stain doesn't soak very well into these phospholipids. So these guys end up that lighter pink color because they don't soak up as much of the stain due to this outer cell membrane. Now, the outer cell membrane also has a special structure found in it called lipopolysaccharide, or LPS. And we'll come back to that here in just a moment as to why that's kind of important, but it's just another molecule that you can find in that outer cell membrane. What's unique, though, is that it's only found in gram-negatives, right? So only gram-negative cells are going to have these lipopolysaccharides. Now, to continue on with external structure, you also kind of can have additional layers outside the cell envelope. You also can have particular appendages extending from the cell envelope. So in terms of extra layers, some bacteria will have a capsule, which is a carbohydrate layer outside of the cell envelope. This is really hard and really tough and very protective for the bacteria. So that can be bad for us, right? If you have a bacterium with a solid capsule, it's a lot harder to kill that bacterial cell, and it could be a much more pathogenic or disease-causing agent. I often like to think of a capsule on a bacterium like an M&M, right? It has that hard candy coating around it, or we're approaching Easter season here. You know, those robin's egg candies you can get that have that hard shell coating on them. That's kind of like what the capsule is for a bacterial cell, if they have one, but they don't all have capsules. Another possibility is something called a glycocalyx, which is kind of fun to say. Glycocalyx is a slime layer. So it's also carbs, but it's not so hard. It's just sort of like this goopy jelly type material that some bacteria will release, and they might coat themselves with it. They might coat themselves and other bacteria with it as well. And this is also protective and adherent. So it's protective, protects them, also helps them stick to stuff. Glycocalyx on a bacterium is bad for us, right? Because if they can have an extra layer that helps protect them and helps them stick to you, like maybe stick to your bones or stick to your skin or whatever, that's going to be tougher for you to kill, right? So glycocalyx these things are good for bacteria, right? Both of these things, but bad for us because it makes it harder for us to kill them. Now, you can also have appendages that stick off the bacteria. Um, Flagella, we saw, eukaryotes can have flagella too, like a sperm cell has a flagellum, a tail. Bacteria can also have whip-like tails. Theirs rotate like a propeller, kind of spin in a circle, really can allow these guys to propel themselves. So there are some bacteria that can actually like burst through your cells along your gut tube or along your intestinal lining if you get them. Now those are rare bacteria, so don't be afraid of that, but in some remote places of the world, they'll get these infections of the reproductive system or of the digestive system, and these bacteria will be bacteria that are found in the water usually. They'll swim into you basically, and then they can propel themselves so forcefully they can basically burst your cells by running into the walls of your tubes. Now they also have the potential to have something called a pilus. A pilus is their sexual appendage. So a while back we saw a picture of conjugation, the bacterial passing of DNA. They do so via a pilus, right? So a pilus is kind of like as close to a sexual appendage as they could have, just forms a bridge between two cells. Now, sort of the last piece to bacterial structure is to talk about bacterial shapes. We saw the three basic shapes here when we did our microscopy lab way back in maybe October, I think. But now just a little extra detail, some more pictures of them here. Remember, cocci or coccus shaped is round or spherical, but then we want to add some prefixes too, because there's prefixes that tell you more about the bacterium. So if you have a diplococcus or diplococci, think two. So these would be pairs of bacteria, or diplococci. Streptococci, there's a lot of different streptococci out there. We're all familiar with a certain one of them, which is Streptococcus pyogenes, which causes strep throat. There's a bunch of other strep as well. And all that means is if you are a streptococcus, you're round bacteria and you come in chains, like you see here in this picture. Now then we also have the staphylococci. Again, most of us are familiar maybe with one of these called Staphylococcus aureus, but there are lots of types of staphylococci, and if you are a staphylococcus, it means you have round bacteria, and they are clusters of round bacteria. So a lot of times we say grape-like clusters. This is a picture of Staph aureus, these little grape-like clusters. So cocci means round, and then we can put a prefix in front of that to tell us even more about it. So round pairs, or round chains, or round groupings or clusters. Second thing we learned before was bacillus. The bacilli are rod-shaped, or sometimes called pill-shaped. Here's a picture of a very traditional-looking rod-shaped bacillus bacteria. This was called Bacillus cereus. You don't have to know that. But and then the last big group are called the spirilla, that you might remember. The spirillum-shaped bacteria, which have this weird spiraling look to them. They're kind of cool. But those are the spirilla. However, I didn't tell you something before, and that is that a lot of bacteria that we're aware of, we actually refer to as being pleomorphic. And pleomorphic means they don't have a real true shape to them, right? Might be kind of oblong, might be kind of boxy, might not show up in groups or clusters or whatever, especially. So a lot of times we don't have a real shape to classify. We just say that they're pleomorphic. Okay, so now let's take a couple minutes to talk about how bacteria make you sick. This was one of our pre-test questions that we saw, is how do bacteria really make you sick, and how do viruses make you sick? As we talked about before, a lot of bacteria are actually called normal flora. They're supposed to be there, right? They help you in ways, or they don't really help you, but they don't hurt you either. Some bacteria, though, can be pathogenic, or the other term we use is virulent. So virulence is an important vocab word. This is the ability of any microorganism to cause disease in someone. So if you have something that's high virulence, it means it can really make you sick. If you have something that's low virulence, it could make you sick, but not as likely. And then if you have something that lacks virulence, then it won't make you sick at all. Now, Bacteria can be virulent for a couple different reasons. Actually, several, but we want to look at two of the ways that bacteria are virulent. So, two of the ways that bacteria can hurt you and make you sick. The first one is what I call sheer numbers, and the second one is toxins. So we'll start with sheer numbers. Again, this is kind of my term for it, but this just means lots of them, right? Bacteria can multiply rapidly inside of an organism. Remember, when we did our 5-second rule lab, we made those plates and we put them in the incubator so it stayed kind of warm and kind of moist. Think about the inside of your body. It's warm and moist, right? So those are the conditions that bacteria like, and so they can multiply by the millions, just overnight even, inside of you. Now, bacteria tend to grow in colonies, and we'll see those on our plates that we grow for the 5-second rule and with another lab that we do in this section. And colonies can be really small or they can be really, really big. And sometimes when we look inside of an organism, you can develop a really large colony that begins to almost act as though it's multicellular, meaning that the cells sort of cooperate. The cells kind of work together with each other. And when this starts to happen, we call this a biofilm. Now, when a biofilm develops, it starts to kind of have these cells that work together, and they also generally will form a slime layer. So they'll have that glycocalyx we saw a couple slides ago covering the whole colony, and that colony becomes really hard to kill. Now, if that colony starts to grow on a part of your body or in a part of your body, it's going to take up space, and it's going to get in the way, and it's going to disrupt your normal tissue functions, almost in the same way that we talked about with cancer. So if you get a really bad infection in your lungs, it's just that you got a ton of bacteria in an enclosed space, right? If you get a really bad infection under your skin's going to puff up. Now, we'll talk later that a lot of that has to do with your immune system, but part of that is also because there's just a ton of bacteria under there. So the first way that they can really affect you is through these sheer numbers. Now, this is a picture up here of a biofilm in the body. This is a bacterium called Serratia, and this picture is of the bacteria growing on an artificial hip. Okay, so if you had a hip replacement as an older person and they didn't do a nice job and keep everything clean and sterile when they did that surgery, you could potentially grow a biofilm on that fake hip, and it would cause you a lot of pain. It would cause the hip to become really stiff, and you probably have to have surgery again. I have a friend of mine. He's actually a really good friend of one of my friends, and he is a priest now. And he's a little bit older than we are, and he had to have his knee replaced. And after about 3 weeks after his knee replacement, he was having so much pain and stiffness and swelling still that they had to do a second surgery, and it was because he had a Serratia infection of that artificial knee, and so he had to have another one done. So it can be really nasty inside of you. Now, the other way that bacteria are virulent, which is really more of the effects that you see in someone that's sick, is through toxins. Right? So essentially poisons created by a living cell. Now, there's two classes or groups of toxins that bacteria can make. The first kind is the most common kind, and that's called an exotoxin. Exo, outside, right? So exotoxins are released by gram-positive and or gram-negative bacteria. Any kind of bacterium can form a toxin, can form an exotoxin, which is usually a protein, and that gets released. These might weaken or damage your cells, might cause your tissues to come apart a little bit. And then what's most important, and we'll talk about this more later, is that they can cause a reaction by your body. So the immune system might react to toxins, and sometimes those reactions might not be a good thing for you, or at least might not make you feel too good. So we'll talk about that more later. The second thing that is possible is called an endotoxin. Inside, right? Endotoxins are built in to the outer structure of the bacterium. So it's almost like touching those bacteria becomes a bad thing that could hurt you. These are only found on gram-negatives, and these are the lipopolysaccharides, LPSs, that we mentioned a few minutes ago. That outer membrane on a gram-negative bacterium will have these LPS built in, and if your eukaryotic cells interact with or touch those LPS, that can also damage the eukaryotic cell and potentially kill it inside your body. So bacteria are in your body, but they're not inside your cells usually, right? They're floating around your bloodstream, they're just underneath your skin, they're inside your gut tube or wherever else they might be. Now we get to switch over to viruses, which I think are pretty cool. Viruses are classified as a microorganism because it is microscopic. Viruses pretty much across the board are pathogenic. Okay, so really we don't know about a virus unless it does cause a disease in you. So viruses are pathogenic and have virulence. These are technically not cells, as we've talked about a little bit already. They're not really alive. They don't meet all 8 characteristics of life, but they do meet some of those, right? So they are going to have some characteristics of a cell, but not all of them. So that's why I have cell up here in quotes, because it's not really a cell. Now what a virus is a little bit of genetic material, okay, surrounded by a protein capsule. And that's it. They don't have any organelles. They don't have any other proteins. They don't have any other, like proteins that would do stuff inside the cell. Maybe a couple of enzymes, depending on the virus you're talking about, get carried. But otherwise, it's just some DNA or RNA, as we'll see, surrounded by some protein. That's it. The outer portion of a virus is protein, and it's called a capsid. So capsids really come in 2 shapes. Capsids can be icosahedral, which is what this is. And so put on your, think like a soccer ball, but each little piece of it is a triangle. So it forms this kind of geometric shape. You can have icosahedral capsids, or you can have helical capsids. It's kind of weird to think about, but a helical capsid would be coiled like DNA or like a spring. So then inside of that, almost like this is a tube or something, you could have the genetic material. If a virus has a helical capsid, it also usually has another coating to it, so it ends up being sort of a spherical looking shape. Okay, so it looks more like a round virus, even if the capsid itself is helical. Now then, viruses come in 2 types outside of that. So you got genetic material, you got a protein capsid. Some viruses are naked. It's a weird term to use, I know, but some viruses are naked, meaning they have their genetic material, they have their capsid, and that's it. Other viruses, however, are called enveloped or enveloped viruses. These viruses have their genetic material, a capsid, and then a layer that is a lipid bilayer membrane. Now, viruses themselves cannot make lipid bilayer membranes. Remember, they're protein and genetic material. They don't have lipids and things that you need to make this. So, a little bit rhetorical here, any guesses as to where they get this envelope? A virus that's enveloped is wearing your skin, right? It's wearing the membrane of whatever kind of cell it infected. Now, this also might allow you to start to understand why viruses can be so dangerous and why viruses can be so hard to kill. Right? Maybe when you were a little kid, you heard the little story of the wolf in sheep's clothing, right? If a wolf dresses up like a sheep, it can sneak in and live amongst the sheep and then attack them more easily. If a virus is coated by your membranes, it can sneak around your body, be mistaken for one of your cells, and can do a whole lot more damage that way. So a virus that's enveloped has a membrane layer around it that it steals from its host. Now, most cells have a couple types of genetic material, right? We've talked about this year already. You got DNA, maybe use some RNA to help you express your DNA and those kind of things. Viruses don't do that. Viruses have one type of genetic material, and they are incapable of reproducing on their own. So they have some genetic material, but they can't replicate that genetic material without some help. Because of this sort of one type of genetic material being present, this is how we tend to classify viruses. We group them based on this. So some viruses have double-stranded DNA, and so that's one group that we look at. Some viruses have single-stranded DNA. That's weird, right? It doesn't appear in eukaryotes, but you can have single-stranded DNA viruses. Some viruses we call positive RNA viruses. They have RNA as their genetic material. They do not have DNA. And that RNA in these guys is just like mRNA structurally. Then the last group of viruses are the negative RNA viruses. Now again, these guys have RNA as their genetic material, and we're not going to worry too much about what this negative means, but you can think of it kind of like their RNA is not quite like mRNA. It would need some kind of structural change to really become like mRNA. So that's why we call it something different, negative RNA. So you got 4 groups of viruses: double-stranded DNA, single-stranded DNA, positive RNA, and negative RNA. So we'll look at those groupings some when we actually look at some specific viruses later. So here's just some pictures. This is from the textbook that Dr. Wong and I wrote. Some pictures of some viruses. You rarely see these guys because they're so tiny. You don't usually see actual pictures of these like under a microscope like you can bacteria. So these are just sort of like, computer images of what scientists are pretty sure these things look like. The first one up here is a bacteriophage. We saw this on the very first slide of these notes. This is actually a virus that infects bacteria. So it won't infect you. It doesn't bother a eukaryotic cell at all, but it infects bacteria. The second one, I'll give you a second, everybody make a guess. This is actually the common cold. And we'll define that a little better soon enough. But this is a cold virus. Okay, and give you a cold. Most of us get these at least once a year. Third one. What do you think it is? You were totally right. This is rabies. Alright, so rabies, which more often infects wild animals than certainly humans and domestic animals. But rabies is a virus that actually gets into your brain, makes you kind of go nuts, and also increases aggression for some reason. So we see this sometimes in some wild animals, and they might cause them to attack other animals or people even, or something like that. Rabies is hard to transmit to humans, so while there have been cases of bats and cats and dogs and stuff getting rabies into a human, it's very unlikely. D and E you've probably heard of before. D's a little bit famous. It's HIV. We'll talk about that more later. And then anybody want to guess? There's one big time virus that we haven't seen yet. This is influenza. All right, so this is what the basic structure of what causes the flu here in E. So we'll revisit some of these later when we talk about common infections that you might get. Okay, so the last little topic here is how do viruses make you sick? So we saw bacterial structure and what they did to make you sick, sheer numbers, toxins. Now we see viral structure, and we want to look at how do they make you sick, because it is a different process. And a virus attacks a host organism differently than bacteria do. And actually, we kind of have a better description of how this works, because it is a 5-step process that pretty much all viruses execute when they infect you. A virus is known as an obligate intracellular parasite. Now, this sounds like a mouthful, but you do want to know that term, and you do want to know what it means. Obligate, right? You're obligated to do something. So obligate means must, right? Intracellular means inside. And then parasites, you might remember from when you were a freshman, is some kind of organism that basically feeds off of another organism, right? It harms another organism, lives off of that other organism. So a virus is an invader that must get inside your cells, and then it will use your cells to survive. So this is the major difference between bacteria and viruses in terms of how they infect you. Remember, like I said a few minutes ago, bacteria, they get in your bloodstream, or they're in your mouth, or they're under your skin, but they're not inside your cells, usually. Viruses are. When viruses get into your body, they immediately go into your cells, so they can survive, basically, because they need your cell to keep themselves alive and then to multiply using your machinery. So let's go through our 5 steps here. Okay, number 1, virus is absorbed or penetrates the host cell. Now, the virus might actually get picked up by the host cell on accident, but through endocytosis, if you remember that term from a section long ago. Endocytosis, the cell basically absorbs it. Sometimes that happens, and it gets in that way. A lot of times if it's going to sneak in like that, it's kind of hiding with something else that's getting ingested by that cell. The second option is it could just cut its way right through the membrane. So you might have a cell, and the virus might just approach it and break its way through and get inside there. Now, once it's inside there, it will uncoat. You're like, what? Yeah, the virus will break into your house, basically, and then take off all its clothes. So the virus, if it has an envelope, that breaks away, and then the capsid breaks open. So this virus that gets inside of you, it falls away, so it releases just its genetic material out into your cell. So step 1, get through the membrane. Step 2, release its genetic material by uncoating. All right, step 3. Step 3 is that the viral genetic material joins with your host genetic material. So if it's a DNA virus, right, the DNA from that virus is in your cell, and it will incorporate itself into your DNA. Boom, boom. Okay, so the blue is your DNA, and then this red viral DNA gets mixed in with yours. If the virus is an RNA virus, starts out with RNA, then what it will do is it will turn its RNA into DNA, and then it'll do this, merging its DNA with yours. So this is kind of like splicing, right? It's going to splice the DNA into yours. And if you have to go from RNA to DNA, we do this with an enzyme called reverse transcriptase. And that was mentioned when we did that DNA microarray lab, but we didn't go into it at all. This is just an enzyme that can do transcription in reverse. So it can go from RNA back to DNA. This basically is the step that involves really 2 enzymes that viruses might carry. Right? One enzyme would be reverse transcriptase if it's an RNA virus that needs to do this. The other would be an enzyme that will help it splice, any kind of virus that will help it splice its DNA into yours. Now the reason it's going to splice its DNA into yours is so that step 4 can happen. And that is that your cell will synthesize and assemble viral products. Yep. Weird, huh? So your cell will express viral DNA. Right? It'll turn that into proteins. Your cell will replicate that DNA to make more DNA. And so what you've basically done is you've created more viruses using your cell. Right? So that's why this is a parasite. Right? It gets in there. It uses the machinery of your cell, your ribosomes, your DNA polymerase enzymes, whatever you've got, to build viral proteins using the host cell. So it needs you to reproduce. Step 5, it gets those new viruses and it releases them, right? Or you have what's called a latent infection. So let's start first. If the cell is going to release those new viruses, which is what happens when you get a bad infection and you're getting this bad cold, it's because this virus is multiplying inside of your cells. And then they release. Now, they could bud off of your cells, which is what you see here on the right. This is where it's almost kind of like exocytosis, right? The virus, the individual virus, buds out of the cell, taking some of your membrane with it. So, alert, this is one of your homework questions. The envelope on a virus, if it has one, is made of the host cell membrane, and it grabs it through budding. If it gets released by budding, then it grabs an envelope made of your membrane. Again, this allows this virus to sort of hide inside of you with a disguise on that is like your cells. Now, the other option is a lot bloodier, and that's called lysis, right, to burst. So what also could happen is we could cause this eukaryotic cell to just explode and hundreds or even thousands of viruses come out of it. this can be really bad if we're talking about one of your blood cells or something, right? It bursts and 1,000 viruses are released into your bloodstream so they can go find a new cell to infect. So as you might guess, this can build up really rapidly in your cells, and you can make viruses in a hurry. Now, the other option than release is called a latent infection, which means the virus just kind of hangs out inside your host cell and waits until later. This is creepy, right? Creepy to think about. But basically, rather than make a bunch of new viruses and let them loose, it makes viruses and they just kind of sit and wait. Now, we'll look at one example of this later, but one of the best understood examples of this is a connection that you might kind of be aware of, which is chickenpox and shingles. Chickenpox and shingles are actually caused by the same virus. The first time you get it, you get chickenpox. Then it actually hides in your body, and we'll talk about this more later, but under the right conditions, it can reinfect you, basically from the inside, and give you a different kind of symptom, which is a rash that we see in shingles. So, this 5-step process, right? Get into your cell, uncoat, incorporate into your DNA, use your cell machinery to make new viruses, and then release is a way that a virus can quickly multiply inside of you and quickly damage you, right? If it destroys every cell that it invades like that by lysing or bursting out, that can start to damage you in a hurry. Now here's our last slide. I know this has been kind of long. This is just an overall image of what we just talked about there, the path of virulence, the way a virus infects you. Don't get too worried here about the different numbered steps, because I know we just talked about 5 steps and this has 10. But this would be how an HIV virus would infect you. Basically, it'll incorporate into your cell and it will uncoat, releasing that genetic material. Now, HIV is an RNA virus, so this step is that reverse transcription step. Then it's going to incorporate that DNA into your DNA. It's going to use that to, and this is showing 2 different pathways, but it's going to use your cell to make new viruses, essentially. Let's go up to here. And then those viruses can bud out of the cell, picking up some membrane from the cell and going on to infect someone else now with this new virus. So that covers all of the background about viral structure, bacterial structure, and then how they infect you. So now we move forward into the immune system of the human, and then also what I think is the fun part, identifying these different infectious diseases.